No te veo respirar, no te veo respirar. It's because she went to me, that's why I didn't say
I spoke to Martin half an hour ago, so he knows. But very nice to know the room number. Because I had to email you earlier. Yes, three different rooms. 14543. Yes.
It says 24 and these are 4. Okay.
Right. Thank you. So I'm sorry for running late. It's my pleasure to introduce Natalia's confirmation seminar. Natalia Andrade is from. Thank you. It's from Ecuador. She's been working on soft corals for some time, octocorals for some time. She did an honours project on Pacific Gorgia, co-speciation plasticity in another octocoral in Ecuador. She's very keen on pursuing what is essentially revisiting what was a very cool area of science in the 1980s. That's kind of molecular, that's the chemical ecology of allelopathy between soft corals and hard corals. This was a subject that was explored a lot in the 80s by natural products chemistry, but there were big limitations on the technology. Now with modern approaches to both molecular genetics and chemistry, it's possible to kind of revisit this area. So the area she wants to work in is a really cool one, a really interesting one, and it's really kind of open for all kinds of novel things. She's been with us for almost a year. She has an Ecuadorian government scholarship, and I'm looking forward to hearing about the latest with Lobophytum. Thanks, Natalia.
Thank you, David. So I'm going to present you my research project for my PhD, that is the immunity and secondary metabolites in the soft coral Lobophytum pauciflorum and the effects of ocean acidification and competition. I'm going to give you a background of my research, then I will give you a detail in this chapter where I will explain the aim, the experimental methods, and the results today when it is the case. And finally, I will give you some explanation about my progress, the timeline, outputs, and budget. As a background, I'm going to talk about three important topics, that is coral competition, immunology in coral, and the effects of ocean acidification. Then I will explain to you how my project addressed these gaps of knowledge. So in competition is a really important interaction that it happens in all ecosystems. In coral reefs is especially important because the species fight for space. Corals, especially that are sessile organisms, are really affected by the lack of space, so competition is a really important part of their biology. They have been dealing with this interaction the whole of their evolutionary history, so we think that this interaction is one of the factors that have shaped coral community that we observe nowadays. Competition can happen in two instances: when the corals are in contact and when the corals are in distance. In both of the cases, they trigger defense mechanisms to fight against the opponent. When they are in contact, we know that these mechanisms are triggered by an immune reaction that is self and non-self-recognition. But this immune reaction is not just a simple self and non-self-recognition; it's more specific than that. But we don't know how it works. In this example, we can see two different species of corals touching each other but not reacting. In distance, we don't know what is triggering these defense mechanisms. So I'm going to give you some examples of how the hard coral defends himself against competitors. First, in this first picture, we can see here that there is some mesenteric filaments that they produce to reach the opponent and digest it externally. Then they can also overgrow the coral, the other coral, as we see here, a hard coral that is involved in a soft coral. So it will eventually involve it completely, stopping the light to come, and the soft coral will die eventually for the lack of light. Then in this picture we see a branching coral growing apart from the brain coral. When the researchers made a close-up of their polyps, they found these sweeper tentacles that are feeding tentacles modified to fight. So that is why the hard coral is growing away from the brain coral, because the brain coral is fighting it. The soft corals can also overgrow the hard corals as a competitive mechanism. As we see here, there is a branching coral that is being involved by the soft corals. Another important mechanism that have soft corals is allelopathy. Allelopathy is the releasing of chemical compounds in the water flow that will kill the opponent or will stop their growth. We can see that there is some bleaching of this is a soft coral and this is the hard coral, and we see some bleaching in the area that is next to the soft coral, but also bleaching in the area that is away. So we think that this is because of the secondary metabolites that are these compounds that I'm talking about. But what are secondary metabolites? In the early of the studies of these compounds, they found out that they didn't have a directly effect in the development of growth, but that's why they call them secondary. But then they discovered an important ecological function of these compounds. They can have really simple or really complex structure, as this component that is a secondary metabolite that has at least 115 carbons in his structure and is produced by a marine organism. In the sea, we can find most of the secondary metabolites found in sea are terpenes by the composition of the molecules. There are several studies that prove that this allelopathy exists. In this study by Samarco and others, they put two soft corals in contact and not in contact with two hard corals. Here we can see the percentage of mortality of the hard coral. They found that Lobophytum pauciflorum has a big effect in Poraides cylindrica, but this was not the case in Pavlona cactus, as we see there are different mortalities. Then in Sinularia pavida was not as deadly as Lobophytum pauciflorum when it was in competition with Poraides cylindrica. And finally, we found that not all soft corals can fight not in contact to his opponent. What made us think that there is some species-specific effect of soft corals on hard corals. Additionally, secondary metabolites has also different toxicities. In this experiment by Acerte and others, they use unfertilized and fertilized eggs of a hard coral, and they put it in presence of the components of only one soft coral. In these graphs, we see the success of fertilization and the success of the vitality of the egg. When they put the first compound that was flexibility, they saw that there was an effect in fertilization, and this was also seen when their eggs were already fertilized. This was not the case with the other compound, DHF, where this one has no effect in fertilization, but once the eggs were fertilized, the eggs had mortality. So this let us think that secondary metabolites from the same hard soft coral have different effects in their opponents. So we know that competition makes the soft coral release secondary metabolites, and there is some studies that suggest that soft corals fight pathogens also with secondary metabolites. But what do we know about the immunology and how the immunology works in soft corals? First, the immunology in these animals have been studied because to understand the evolution of the immune system. We know now that there are some genes present in humans that are also present in corals. In this study, where they, in this phylogenetic distribution of an immune gene family, we can see that the red names are the ones that possess this immune gene family. We see that the presence in Homo, that will be humans, and in Acropora, that will be corals. That suggests that there is an ancestral, common ancestral that possesses this immune gene family. Another important topic why people is studying immunology in corals is coral diseases. But most of the studies have been done in hard corals and not much in soft corals. There is a specific study about the genetic expression when the hard coral is in a bacteria infection, but there is no known about the bacteria infection in soft corals. Nevertheless, we have some studies that are recent and but we lack information about these organisms. Coral diseases affect hard corals and soft corals, and this outbreak of diseases have been occurring because of the rise of temperature. That is one of the stressors of climate change, but we don't know what is the effect of another stressor that is ocean acidification in the immunology of hard corals. So ocean acidification is the rise of the concentration of CO2 in the water that will make the pH, down the pH in the water. There is some studies that suggest that there is effect in the metabolism of the hard corals and in the calcification process when they are in these highest concentrations of CO2, but we don't know what is happening in the immune system when they are in this stress. There are other studies in higher organisms as Drosophila and Mammalia where they found that CO2 has an effect in their immunology. So to sum up what I have said, we know that competition triggers the production of secondary metabolites, but also a reaction, immunology reaction. So we would like to know if the secondary metabolites are the same. Then the gene expression when they are in competition is unknown, as well as the gene expression that happens when they are in an immune reaction. So we would like to understand if there is any similarities here. Finally, there is no knowledge of how the soft coral react to a bacterial infection. That is another gap of knowledge in this topic. Finally, we don't know what is happening or competition or immunology with ocean acidification and how this stressor will affect the corals and their interactions. I will address the different gaps that I mentioned with first the gaps about competition and secondary metabolites with my two first chapters, but I will try to characterize the secondary metabolites, the nature of the secondary metabolites in this soft coral, specific soft coral, Lobophytum pauciflorum, but I would like to go deeper and understand if there is any specific gene expression when the soft coral is in competition with two hard corals. Then, for the gap of immunology in soft corals, we will try to understand which genes are involved in an immune reaction of these animals. And finally, to understand what are the effects of ocean acidification, we will create a scenario with rays of CO2, and we will also have a genetic approach to this chapter. My model species for this research are Lobophytum pauciflorum, that is the soft coral that is present in abundance in the area that I'm going to work, that is Orpheus Island. We have already the transcriptome of this organism, so the data analysis will be easier. Also, it has, the literature suggests that has really potent secondary metabolites, so that's why we choose it. Acropora millepora will be one of the hard corals that we use for the competition between soft coral. He's a really well-known hard coral. We know a lot about his genetics and ecology, so to know a little more would be good. And for Iris cylindrica, we know that this hard coral is sensible to the secondary metabolites or Lobophytum pauciflorum, and this hard coral is in danger in some localities, so it's important to know more about its ecology too. In my first chapter, where I will isolate the secondary metabolite as possible, my aim is to establish the nature of these secondary metabolites. For this, first I'm going to extract the whole secondary metabolites or my sample, that will be Lobophytum pauciflorum. Then I will test the effect of this crude extract in the two hard corals. Once we know that the effect, we will separate the different compounds, so the different fractions of this crude extract, and we will test again the effect of these different fractions in the hard corals. Finally, we will try to identify the strength of toxicity of each of the fractions selected. This is a first step that I already done. I collect the sample and I freeze dry it. I extracted the secondary metabolites with dichloromethane and analyzed the crude extract with NMR. I also test the effect of the crude extract in the hard coral last month. In this experiment, I took 3 colonies of Iris cylindrica and 3 colonies of Acropora millepora. Little pieces of the 3 colonies of each species were put in small tanks, as we see in the picture. In the treatments, I put a concentration of the crude extract of 1 microgram per milliliter, and the crude extract was dissolved in isopropanol. That's why in the control we use isopropanol to detect if there was any effect of the solvent. The data collected here was RNA sample of each piece of coral, tissue sample for histology. I took pictures to visualize the effect, and I measured the photosynthesis efficiency with the diving pan. The results of this experiment, as we see here, there are the two hard corals in time 0. We can observe the polyps that they are alive and happy. And after 24 hours of the treatment, the hard corals die and there is almost no polyp left. So we can assume that the crude extract has an effect in the hard corals. Now what we have left is the separation that we are going to do it by column chromatography and the analysis of each fraction in NMR. If we will need some extra purification, we will use HPLC. After we have the different extracts, we will repeat the experimental design that I did already for the crude extract, but with the different fractions. Again, with a high concentration that is higher than what the literature suggests. The same data will be collected in this space. Finally, for this chapter, we will test the toxicity of the strength of the fraction that has the effect of the hard coral in the hard corals. We will choose a high and a low concentration. This concentration will be determined first in a pilot when we will prove a broad of concentrations to understand what is high and what is low for the compound. We will use again 3 colonies of each hard coral, and the same data will be collected. Now that we know, or that we that I explained how we will identify the nature of the secondary metabolite, we want to know a little bit more. So we will do the transcriptome analysis of the soft coral when it is in competition with 2 hard corals. The aim is to determine the genetic basis of this mechanism of defense and also the production of the secondary metabolite. We will put the soft and the hard coral in a competitive scenario, which means in the same tank, and after the treatment we will do the transcriptome analysis. As I told you earlier, we have knowledge from the literature that Porairis cylindrica is sensible to the secondary metabolites of Lobophytum pauciflorum, so we know that they interact. But we didn't know what happened with Acropora millepora and Lobophytum pauciflorum. So we made a pilot experiment to identify this interaction. In the pilot experiment, we choose just 2 colonies of Lobophytum and 2 colonies of the hard coral, and we put the little pieces next to each other but not touching. We collect samples of RNA of each coral. I took pictures weekly and made a lot of observations. As a result, we found that the novins that were in presence of the soft coral start producing these secondary metabolites. These mesenteric filaments, that they are triggered when the hard coral is in competition. As we can see here in this picture, they release the mesenteric filament in the water to try to reach the opponent. We found additionally that 67% of the novins that were in treatment produced these mesenteries more than once in the duration of the treatment. Now what we have left for this chapter is to do the competitive experiment where we will put the 2 species of hard coral with the soft coral. We will choose 5 colonies of Lobophytum because we are going to work with genetic expression, so that will help to have a better data set. We will take the same samples as in the other experiments, but additionally we will take little pieces of the soft coral to do the chemical analysis and understand which secondary metabolites are producing. Finally, the transcriptome analysis, we will have first to do the RNA extraction. We will use the RS sequencing method, that is the construction of libraries and sequencing, to have a data set that helps us to identify the target genes, and these target genes involved in competition will be validated in qPCR. So now that we understand which genes are involved in competition, we want to know which genes are involved in the immune system and if they are the same ones that we find in competition. For this, we are going to put Lobophytum in an immune challenge, and after the treatment, we will do the transcriptome analysis for this chapter. We are not going to infect the soft coral with bacteria. We are going to simulate the infection with a molecule that is muramyl dipeptide that is present in the membrane or gram-negative and gram-positive bacteria. So we will inject the molecule in the soft coral to simulate this infection. Again, we use 5 colonies for the same reason. The data collected here will be the same that we will collect for the competition experiment, and the transcriptome analysis also is the same that I explained in chapter 2. For chapter 4, that we want to understand the effect of ocean acidification in coral competition, we want to investigate what are the effects of this environmental stressor in the defense mechanism and the production of secondary metabolites. So for this, we will put the soft coral and the hard corals again in a competitive scenario, but now we will raise the concentration of CO2 in the treatment. After the experiment, we will do the transcriptome analysis for the samples. In this experiment, we will use a control. The control will be the seawater that is pumped from the station at Orpheus Island. And in treatment, I will use 1000 ppm of concentration of CO2. That is what is predicted for 2050. The data collected will be the same in the other chapters, as well as the transcriptome analysis. So all this experimental design that I have done and all the project will help me to identify the secondary metabolites involved in competition, as well as the genetic expression in this scenario. Secondly, we will also understand the immune response of the soft coral to an infected bacteria. And finally, with the final chapter, we will understand a little bit more about the effect of ocean acidification in coral competition. So now, my progress until now, in general, I already did some compulsory workshops, or all of them. I'm attending a SCIP. I took the subject molecular genetics, that was one of the requirements. In lab work, I did the extract of the secondary metabolites of the soft coral, and in tank experiments, as I already mentioned, I did the pilot experiment to understand the interaction, and I test the effect of the fruit extract in the 2 hard corals. In my timeline, as you can see here, I'm planning to work in 2015 in my fieldworks to do all my experiments, and to have in the mid-year to have the data to do the extractions and preparations for the sequencing, and finally to analyze the data and write the thesis in 2016. We aim to publish at least 1 paper for each chapter in different target journals. this is the budget of my project. Most of them, the most costly is the transcriptome analysis, as some of us know. And I would like to thank first the Senacid, that is the scholarship that I have. My advisory panel have helped me a lot. All the members of the lab, especially Vic and Anthony, and all my friends, especially Diana, Annie, Nicolas. Thank you.
Natalia, thanks very much. How do you quantify the secondary metabolite?
So the quantification of the secondary metabolite, we are going to do it with NMR2. So with the NMR and knowing the weight of our sample originally, we can measure the concentration that we see in the above of the curve. So that's how we are going to quantify.
So you're adding consistent amounts of the secondary metabolite in doing your experiment?
Sorry?
Are you adding consistent amounts of the secondary metabolite in each of your experiments?
Sorry, when I'm doing the experiment to know the effect, okay. To do the effect, no. What I did was I keep the tanks without water flow and just air. So air bubbling. So first I did some trials to see if the corals will survive properly without water flow, because it's an issue that not to have. But then they live happily for at least a week without water flow, and then we start seeing some effects. But that's how I kept the concentration constant in the experiment without adding that.
That's it.
Yes.
I have a question. With the CO2 experiment, is there a reason why you're only using 1000 ppm and not other measures?
Yes, first because it's a big experiment that I will have to do, and it's really costly. So we have to choose one of the concentrations. So I choose one of the highest one to know really if there is going to be an effect or not.
Looks like we're done. Okay, so thank you, Natalia, again. I'm sure there's lots of interactions that people have, questions that people will ask informally afterwards.
Okay, thank you.
Thanks.
Your questions, sorry?
Estuvo bien. No puedo.
Excellent.
Come on in.
Now we have. Yeah, of course. I will tell you. Don't you want to? Probably I will do something. I also, I don't know how to turn off. I will text you. We have to leave. I think that you will. Muchas gracias. Tenemos que salir.
Ya, pero saca la...
Vos no habías visto mis presentaciones. Solo déjalo. Ya
¿Y tu pendrive?
Ya le pasé. Yes. Are you going to meet here or in
No,
another?
we'll go to David's office, I think.
Ah.
So,
David's,
yeah.
So I'm going to explain you. This is the report that you have to fill with all of them of the presentation.
Yeah.
Okay. from the writing and the proposal that the advisory panel have to give. And this is the checklist, so I'm not going to give you this because
Yeah.
the reader is okay.
Alright, thanks.
Okay.
Well done.
Thank you very
It was
much.
much
No
better.
worries.
It was much better. It was
Yeah.
great. No,
Okay.
I don't anticipate any. Right, you know, somebody's left water here.
I think it's Susie's. ¿Qué más? Pensé que se iban a reunir aquí y les dije a todos salir corriendo.
¿A quién?
Al resto.
Ojalá.
Ay, qué bestia, qué nervios.
No vale, pero lo hiciste muy bien.
¿Te parece? Me trabé full.
No vale. Bueno, sí, no sé, no me di cuenta.
Ay,
Bueno, tengo que ir al lab. Te voy a dejar algo en tu cuarto.
ay, muchas gracias. Muchas
¿Qué vas a
gracias.
hacer? Regresas a tu casa, me imagino.
Ay, ahorita sí, no sé qué voy a hacer, nunca planeé esto.
O tienes que esperar que te hagan
Creo
una
que tengo
nota.
que esperar.
Que te vayan a buscar a tu oficina.
Me imagino, porque se fueron a la oficina de Baby Yellow. Chuta, nosotros fue al migrante con la Coca-Cola aquí.
Celular. ¿Tu pendrive lo
Sí,
tienes?
ya la cogí. Pero sigue grabando y...
Parece, sí. Bueno. Ahora te veo.
Gracias.
Vamos a la oficina. Ay, una cosa